Hey guys, welcome back to my channel. So today I'm going to continue the Pharmacology 1 4th semester question and answers as per RGUHS. So um, in the previous video we have started the 10 marks for the second unit, that is general pharmacology, and this is a continuation from the 6th question. So I'll start from the 6th one, that is what are the different types of drug interaction and explain the factors affecting pharmacodynamic drug-drug interaction. So the different types of drug interactions, you have to refer the question number 5, and coming to the factors affecting for pharmacodynamic drug-drug interactions are, in that the pharmacodynamic drug interactions, these are those in which the object drug at its site of action is altered by the precipitant, and such interactions may be direct or indirect. So there are two types of pharmacodynamic interactions, in that number one, direct pharmacodynamic, and number second, indirect pharmacodynamic. So for that, the first one, the direct pharmacodynamic is, these are in which the drugs which are having the similarities of opposing the pharmacological effects are used concurrently. So there are three consequences for the direct interactions are antagonism, addition or summation, and number third, that is synergism or potentiation. So number one, antagonism. So the interacting drugs having opposing actions. Example, acetylcholine and noradrenaline has opposing effect of heart rate. Number second, addition or summation. So the interacting drugs have similar actions and the resultant effect is sum of the individual drug response. So the example, CNS depressants like sedatives and hypnotics drugs, etc. So number third, that is synergism or potentiation. So it is an enhancement of action of one drug by another. So the example, alcohol enhances the analgesic activity of aspirin. And number second, that is indirect pharmacodynamic interactions, so in which these both the objects and the precipitant drug have unrelated effects, but the later in some way this alters the effect, and for the former, consequences of drug interaction, for major they are life threatening, for moderate deterioration of patient status, and for minor little effect. So reducing the risk of drug interaction, identify the patient risk factor, take through the drug history, be knowledgeable about the actions of the drugs which are being used, consider the therapeutic alternatives, and avoid the complexest therapeutic effect which is possible, educate the patient, and monitor the therapy. So question number 7, explain the different preclinical evaluation phase in drug discovery. So the preclinical evaluation phase are the non-clinical trials and are laboratory tests for a new drug substance or a medical device, usually which these are done on the animal subjects to see whether the treatment really works on it, and if it is safe to test on humans. So the main goal of preclinical studies are to determine a product's ultimate safety profile. So the product may be include new medical devices, drugs, gene therapy solutions, etc. So after identifying a compound, it is tested on animals to expose the whole pharmacological profile, where the experiments are generally performed on rodents like mouse, rat, guinea pig, hamster, rabbit, etc. After the successful results, experiments are performed on a larger animal like cat, dog, and monkey. So, as the evolution progresses, unfavorable compounds get rejected at each step. So, that the only few out of thousand reach the stage when the administration to man is considered. So there are several steps which involved in doing a preclinical trial are identify a drug target, develop a bioassay, screen the drug in the assay, establish effective and toxic doses, file for approval as an investigational new drug, that is IND. So the types of studies which included a preclinical trials are screening test on isolated organs like bacterial cultures, test on animal models for human disease, general observation test and confirmatory test and analog activities and mechanism of action, systemic pharmacology, quantitative test, pharmacokinetics and toxicity test. For the screening test, these are simple and rapidly performed tests to indicate the presence or absence of the particular pharmacodynamic activity. Example, analgesic or hypoglycemic activity. Number second, test on isolated organs or bacterial cultures. So these are preliminary tests to detect the specific activity such as antihistaminic, antisecretory, vasodilator, antibacterial, etc. And number third, test on animal models of human diseases. So animal models used like kindred seizures in rat, genetically hypersensitivity rats, experimental tuberculosis in the mouse, etc. And number fourth, general observational test, that is drugs are injected in tripling doses to small groups of mice which are observed for overt or hidden effects. So the preliminary clues are drawn from the profile of the effect which is observed. And number 5, confirmatory test and analogs activity. So the compounds which are found active are taken up for detailed study by more elaborate, which is complex tests which confirm and characterize the activity. So on the other hand, the related activities also measures like antipyretic and anti-inflammatory activity and analgesic. So number 6, that is mechanism of action. So the attempts are made to find out the mechanism of action. Example, whether an antihypertensive is a blocker or beta blocker or ACE inhibitor or calcium channel blocker, etc. So number 7, systemic pharmacology. So the irrespective and the primary action for the drug, its effects on major organ systems like a nervous, cardiovascular, respiratory, renal, which are worked out. And number 8, quantitative test. So the dose response relationship, maximal effects and comparatively the efficacy with existing drug is carried out. And number 9, that is pharmacokinetics. So the dose response relationship, maximal effects and comparatively efficacy with existing drug is carried out. And number 10, that is toxicity test. So the acute toxicity, single high doses are given to a small groups of animals that are observed by the for the overt, which is hidden effects and mortality for 1 to 3 days. So the dose which kills 50% animals is called LD50, and the organ toxicity is examined by the histopathology on all the animals. So these whole tests are carried out under the standardized procedure and under the good laboratory practice, that is GLP. And the good laboratory practice applies to the non-clinical studies which are conducted for the assessment of the safety or efficacy for the chemicals to man and the animals and the environment. So importance of preclinical trials to determine the dose, toxic dose, pharmacological action, etc. So it is the requirement to the regulatory body for performing a clinical trials, and as the ethical viewpoint, it is necessary to check the safety of the drug on animals before starting to check on human beings. So to check the kinetic profile of the drug and based on it select the route of administration in humans for the clinical trials. And question number 8, explain the different clinical evaluation phase in the drug discovery. So after the completion of preclinical studies, the IND is filed with the FDA in the United States, so the drugs is continued for the further research and testing in the human beings for clinical trial phase. So the following 4 clinical trial phases are human pharmacology phase, therapeutic exploratory phase, therapeutic confirmatory phase, and post marketing surveillance. So though the phase 0 studies are not very common and are not mandatory as of now, as per regulations, we would describe them too. So the phase 0, that is microdosing studies, recently FDA has approved the testing of small quantities for the experimental drugs in the human beings to understand the path of the drugs in the body. So US FDA designated this phase as a phase 0 in accordance with US FDA 2006 guidelines on an exploratory INDA. So a 0 phase can be studies in considered as a microdosing studies. So these studies which gives no data, known safety or efficacy by the definition. So a dose too low to cause a therapeutic effect, drug development companies and carry out the phase 0 which has the best pharmacokinetic parameters in humans to take forward into the further development. And number second, that is phase 1 human pharmacology phase. So the phase 1 trials are conducted to determine the pharmacologic parameters, initial safety and tolerability of the drugs in human beings. So usually healthy volunteers participates in these trials, but in some special diseases like conditions like cancer, AIDS and similar conditions, the disease population is targeted where the participation of healthy volunteers is ethically not possible. Usually a less number of patients, where generally 20 to 80 are included in these trials. So these studies typically include both single and multiple dose of administration studies. And number third, that is phase 2 therapeutic exploratory phase. So it is an exploration for the primary objective, that is to explore the therapeutic efficiency of the drugs in patients. Once the parametric pharmacology is determined for the experimental drug from phase 1 studies, depending upon the data obtained from the phase 1, and the dose regimen and the dose efficacy is determined in 2 sub-phases. So the phase 2A dose regimen and dose requirement are assessed, and phase 2B efficacy of drug at prescribed dose regimen is evaluated, and phase 2 plays a critical role in the successful drug development process. And number 4, that is phase 3 therapeutic confirmatory phase, where the phase 3 trials are present as a bridge between the clinical research and the application of its experience in the clinical practice. So a product that successfully comes through a phase 2 trial may be put into the phase 3 clinical trial. So the phase 3 studies are most often designed to compare the currently available care treatment with the investigational agent over a large group of patient pool. So in this phase, the safety, efficacy, parameters, side effects, risk and benefits for the investigational product are the main focus. So the phase 3 trials may be double blinded, where neither the investigator nor the subjects know which treatment is being used, or a single blinded, where only the investigators knew which treatment is being used. Once the phase 3 data is obtained, NDA is submitted to the FDA. So the FDA reviews all the data submitted and approves the drug for marketing if the data is found worth. And number 5th, that is phase 4, post marketing surveillance. So the post marketing surveillance research is an important element of commercialization that enables the company to expand the existing markets, where enters to the new market which develop and deliver messaging and that directly compares to their products with the competition and a secure position in crowded markets. So the phase 4 trials are done to determine a drug or the treatment is safe over time, or to see if a treatment can be used in other circumstances. So these studies are often important for the optimizing drug use. So the safety surveillance is designed to detect any rare or the long-term adverse effect over a much layer patient population and longer period of time. So then in phase 1, phase 2 and phase 3 trials. So the phase 4 studies can be result in a drug or device which being taken off the market, or restriction of use could be placed on the product depending upon the findings in these studies. And question number 9, what are detailed note on mechanism drug action? Write a detailed note on mechanism of drug action. So for mechanism of drug action, you have to refer question number 1 from 10 marks. So here we have completed the 10 marks question and answers for second unit Pharmacology 1 4th semester. So tomorrow I'm going to continue with the 5 marks of Pharmacology 1 second unit. So please do like, share and subscribe to my channel.